This recorded session is dedicated to the last part of our module on the endomembrane system. So this is a lecture on the Golgi complex and lysosomes. So for the first parts of module 5, we talked about the general characteristics of the endomembrane organelles. In particular, we emphasize a lot on the endoplasmic reticulum. So we already covered discussions on how proteins are translated in reference to their final destination and in the context of the rough endoplasmic reticulum as an active component of the eukaryotic cell responsible for translation and modifications in the final protein product. So for this component of module number 5, we'll cover the Golgi complex, the processes happening within this organelle, and how proteins are sorted. In particular, um, I don't know, after post-translational modifications, how are proteins labeled so that they can actually be so that they can go to their final, or they can be delivered to their final location, to their final sinks. Okay, so let's first talk about the Golgi complex. So the Golgi complex is a series of membrane bound cisternae. Okay, so similar with the biological nature of the rough endoplasmic reticulum, the Golgi complex looks like a flattened stack, or a flattened stacks, no, of cisterna, which are essentially membrane enclosures possessing space within it. Okay, and if you were to further dissect the Golgi, you can appreciate the different regions of this organelle. There is a cis region, or a cis phase, the cis Golgi network. There is a trans region, or a trans phase, the trans Golgi network. And between these two areas is a medial or median Golgi network. Okay, so the cis phase is the one nearest or nearer to the endoplasmic reticulum, while the trans phase is the region where endosomes, where transport vesicles are emanating from. So essentially, or basically, when it comes to putting a direction, you expect that from the endoplasmic reticulum, the vesicles are entering, or vesicles are expected to enter the Golgi region through the, or to the, I don't know, through the cis Golgi network first, and then travel siya towards the medial, and then eventually it will be secreted out of the complex in the transphase area. Okay, so transport vesicles, they carry proteins, essentially I don't know, most of the time proteins, also some biomolecules such as fatty acids, lipids, from the ER network to the Golgi network, and from the Golgi network to different destinations within the cell. Okay, a little history about this organelle. The name is attributed, or is I don't know, given in honor of the Italian biologist Camillo Golgi, 1898. So um, early 1900s was the time, no, that the organelle was first observed. Further interest actually, I don't know, poured into the study of this organelle. Okay, And if you were to look at if you were to factor the nature of the cell, and you'll come to the realization that um, the microscopic appearance of the Golgi will be different according to the function of the cell. So you expect that cells that are more dedicated, that cells that are dedicated to secretion, to production of secretory proteins, okay, they will have more distinct Golgi complex or Golgi network. Okay, so you can see here these two pictures. One is an artist's rendition, an artist's interpretation of the Golgi network representing or depicting the three regions, okay, the CGN, the medial cisternae, and the TGN. Then here is a electron micrograph of the Golgi stack in an algal cell, in a cyanobacteria. So you can appreciate also the depict the regions in relation to positioning, in relation to the areas that are near to it. Okay, so we can appreciate that the cis Golgi network is nearer to the endoplasmic reticulum compared to the trans Golgi network. Okay. Now, more about these two important areas of the Golgi complex. The CGN, you expect vesicle fusing with it, and the transport vesicles are primarily from the ER, while TGN, which is on the opposite side of the network, the opposite phase, you expect substances to leave this area. No? So from the Golgi, if the protein or the secretory product is labeled to be delivered to another component of the cell, yeah, transport vesicles emanate from the trans phase, carrying with them the labeled protein, the sorted protein, or whatever product is present within the transport vesicle. Now, the function of the Golgi is tightly connected with the endoplasmic reticulum, with a lot of post-translational events happening primarily in the medial component, the medial region of the Golgi network. So a lot of protein processes actually occur. Now, the evidence, some of the evidence that actually point to the idea or the hypothesis that the three regions of the Golgi network actually possess different functions. Some of the early evidences include this electron micrograph showing us an immunochemically stained preparation of the Golgi complex. Okay, so this micrograph shows a Golgi stack in a rabbit kidney cell. The cell section has been stained to detect a special enzyme, a transferase enzyme, and acetylglucosamine, which is important during terminal glycosylation. So you can see the darkly stained area is actually the medial network. So the medial network is the stacks of cisternae that contain a lot of the enzyme, while the cis Golgi network and the trans Golgi network are lightly stained, suggesting that the concentration of the enzyme is lesser in these areas compared to the medial network. Okay, now since we've talked about the the microscopic appearance of the Golgi network, then the areas that we have to I don't know to look at, the three regions. Now, let's now examine some things, or let's now go on to I don't know the discussion about um, how are molecules, how are biomolecules shuttled from the ER through this network. There are actually two standing hypotheses regarding the flow of biomolecules within the Golgi complex. So the first hypothesis is the stationary cisternae model, which states that each component of the Golgi complex is stable. So the three regions are stable areas that possess distinct features, distinct enzymes, distinct membrane proteins, no, within the ano yan, yung membrane nung cisterna, okay? And shuttling of or the flow of molecules such as lipids and proteins are via shuttling via the action of transport vesicles. Not so it is transport the major shuttling elements are the transport vesicles. They allow trafficking of substances through the CGN to the medial region to the TGN. Now, the second hypothesis on how molecules are trafficked and how are they flowing from one component, one region of the Golgi to the other, from one end to the other, is the cisternal maturation model, which states that each part or each region of the Golgi are actually transient. No? They are they are cisterna that actually change their nature as they accumulate particular proteins, particular enzymes within the lumen space that are responsible for carrying out the functions happening in each region. No? So there's maturation of the cisternae from the CGN to the TGN. Okay. Now, in both of these models include, or both of these models, I don't know, let me qualify. Central to these two models are transport vesicles. So both models make use of transport vesicles as important carriers, no, as shuttlers of the molecules from one area, from the CGN to the TGN. Okay. Now, as I mentioned, the Golgi and its biology is tightly connected with the endoplasmic reticulum. With the ER, some of the functions that are shared, or in a way you can think of these functions as as continuous from the ER to the Golgi components, the Golgi complex. So with ER, protein glycosylation and modifications are also carried out in the Golgi. Protein trafficking and sorting, of course. No? We'll talk about more on these functions in a little while. We will dive deep into these functions. No? There are some important mechanisms and elements that we have to cover. And then in continuation or in continuity with the functions and the I don't know physical existence of the ER, the Golgi is responsible for the formation of lysosomes. Then cellular secretion, and other cellular transport processes are also covered or carried out by the Golgi complex. No? Actually, loose yung definition natin ng secretion and transport because in a way, ano pa rin naman siya these processes happen in relation to how or in relation to the ER, in relation to what the ER diverts to the Golgi network. Okay? Now, let's first examine, as promised, no, discuss natin to lahat. Let's first examine glycosylation. No? Glycosylation, we've talked about glycosylation in the endoplasmic reticulum, no? And we've defined it as a process where carbohydrate chains, no, are attached to amino acids of a polypeptide chain. Okay, and we discussed largely about in the previous lecture, previous recorded lecture, we discussed some of the elements that are involved in the process of protein glycosylation. So we can delineate the process, or we can think of the process as divided into two stages. So there's initial glycosylation happening in the endoplasmic reticulum, and there is final or terminal glycosylation happening in the Golgi. Now, the difference is, tatandaan natin, basic difference. In the ER, glycosylation of a protein, of a polypeptide, is more on addition. So you expect sugar molecules, sugar monomers, and branches being added to the protein. While in the Golgi, ang terminal glycosylation is more on pruning, modifying the placement of the sugar molecule of the monomer. So it's more of parang finishing touches pag terminal glycosylation. No? So in initial glycosylation, addition of the monomers. While in terminal, it's more on polishing, final touches. Actually, no, no, very active subtraction, or ano no, cleaving, maraming hydrolases, no, glycan hydrolases that are present in the in the Golgi network, primarily in the medial region, responsible for removing, adding, modifying the location of the monomer of the sugar monomer, or the sugar branch, no. Okay. Now, from the ER, we will remember, or you will remember from the previous lecture, yung discussion natin about the core oligosaccharide, which is essentially composed of different types of sugars, sugars such as glacnac, mannose, and glucose. No, very very evident in a lot of glycosylated proteins, a lot of proteoglycans. These monomers you expect them, no, to be present all the time. No? Mannose, glacnac, and glucose. Okay. Now this figure here shows us just a schematic of what's actually happening within the GC, within the Golgi complex, during terminal glycosylations. As I mentioned, no, a lot of removal and modifications are happening during terminal glycosylation. Now, also, you can think of terminal glycosylation in the in the context of adding in the context of adding important sugar monomers to to glycoproteins. Some of the three most, I wouldn't say most important, but probably you will encounter a lot of times, no, from this point forward. Sugars that are added to terminally glycosylated proteins. Tatlo, no? So we have galactose, we have sialic acid, and we have fucose. Yung tatlong sugars na yan, paulit-ulit ma-recognize or ma-encounter niya sila, probably in your higher medical courses, no? In particular with diseases. Yan. There are some diseases associated to the failure of proper terminal glycosylation. Like for example, cystic fibrosis. No? For the case of cystic fibrosis, it's a problem of sialic acids being present, or being improperly modified, or improperly attached to the final protein phenotype. Okay, so that's just one example. Then there are also instances in which an organism, a phenotype, becomes more susceptible to bacterial infections because of fucose monomers. So yung proteins nila that are possessing fucose monomers, yan. So fucose has been identified to be a common molecular signature that a lot of um, a lot of microbes recognize as a receptor. So imagine if the protein is a surface protein, tapos is a cell membrane protein, no? Nasa plasma membrane siya. So if that protein becomes improperly glycosylated with fucose, so it can be targeted. It can be more susceptible to microbial infections compared to the normal phenotype, to the wild type. Okay? Now, let's review before we look at terminal glycosylation, no? At ano muna, let's refresh our knowledge base on glycosylation happening in the ER. Yeah. Okay, so um, this is just a review of what I recorded last time. Yeah. So and remember that for proteins that are translated or imported to the ER, okay, those proteins that will require um, additional sugar molecules or will require glycosylation. Okay, the synthesis of the glycan branch or the sugar branch will be starting or is expected to start outside the ER cisterna. So it's in the cytosol where the branching of the core oligosaccharide will be initiated. No? Then you also let's be reminded that an important anchor of the core oligosaccharide within the ER membrane is the biomolecule dolicol P. Diba? So dolicol phosphate. Okay. Then enzymes within the ER known as flipases are responsible for retrotranslocating the growing core oligosaccharide chain from the cytoplasm from the cytosolic space to the ER space, to the ER lumen. Okay, and then further additions will happen in the ER lumen, and eventually once that the protein with its added core with its added glycan chain or oligosaccharide chain, sugar chain is finally I don't know finally done with the synthesis, and that's the time that from the ER will shuttle it towards the Golgi complex. In the Golgi complex, as I've already no, no, hinted a while ago, a lot of removals, polishings, uh, no, no, branch modifications happen. So primarily, these things serve very important purposes because the final function of the translated protein will be heavily dependent on the nature of the core oligosaccharide attached to it or on the nature of the sugar chain attached to it. Okay, so some of the uh, things happening here aside from removal of the carbohydrate units include addition of sugars, galacnac, galactose, sialic acid, and fucose. Primarily, these monomers are added in the last areas, no? Yung ano na, from the medial going to the trans, this important monomers of sugars are added. Okay? Eventually, ano na, papalapit dun sa secretion part. No? Palapit dun sa area na they're already ready to go. They're already ready to be uh, they're ready to be um, released from the Golgi region, from the Golgi network. Okay. Now, aside from the addition of sugars, there are also other things that happen within the Golgi compartments, covalent modifications that allow proteins to have specific structure and function after they are released from the space, from the organelle. No? There are modifications of yun nga, existing sugar moieties. There is O-glycosylation. Okay, so you remember the difference between N-glycosylation and O-glycosylation. So N-glycosylation is the addition of the sugar chain to an amide nitrogen, di ba? Yung mga ano natin, asparagine, okay, is an example. O-glycosylation, on the other hand, primarily happening in polypeptide monomers that are made up of that are either serine or threonine amino acids. The addition of the sugar chain to the oxygen atom. Okay, it's O-glycosylation. Yung pa pala, no? Another difference. A lot of N-glycosylation is happening in the ER, while a lot of O-glycosylation is happening within the Golgi, okay, within the Golgi network. Other events such as sulfation, addition of sulfates to the hydroxyl component of tyrosine amino acid. Phosphorylation is also another post-translational modification happening, okay, within the Golgi compartment. Okay. Okay. So this next slide, I think I've mentioned all of this already. Yeah. Okay. So next, let's now take a look. Let's have an illustrative example of how terminal glycosylation occurs in the Golgi compartments. Yeah. Okay. So you can always go back to this figure every time that you want to refresh yourself about what's happening during terminal glycosylation. So let's say that our raw material is an N-glycosylated protein with core oligosaccharide attached to N-glycosylated in its asparagine residue. Okay, so what proteins, what enzymes come into action, come into play? So we have glucosidases and mannosidases. So here in step 1, within the ER lumen, glucosidases can act upon the upon the N-glycosylated protein and can remove those sugars, as well as mannosidase. Pwede rin siyang mag-act to actually remove sugars. So you can see here a change in the oligosaccharide chain, diba? Nawala itong glucose monomers and then nawala yung isang mannose monomer. Okay? Now, within the Golgi lumen, within the Golgi network, CGN to medial, additional things happen. Okay, so here you expect enzymes such as similar with the ones working in the ER. Okay, so you have a Golgi mannosidase working to cleave or to take away the mannose component, the mannose monomer. Okay, so a change in the core sugar chain is appreciated from step 1 to step number 2. Okay, so you expect itong mga ACEs na to, mannosidase, glucosidase, these are hydrolase enzymes. Okay, so they work by hydrolyzing the polymer, okay, the dimer of sugar, to divide them into their simple monomeric components. So these ACEs are responsible for cleaving off the sugar monomer from the branch. Okay, now in this third step, we have the addition of aglacnac, no, an N-acetyl glucosamine sugar, okay, yellow, represented by this yellow component, from a nucleotide phosphate, uridine diphosphate, if I'm not mistaken, UDP, carries with it a GLAC-NAC monomer, okay, and with a transferase enzyme and acetylglucosamine transferase, okay, this enzyme acts upon the edited um, sugar chain, and then ayan, madadagdag na siya. Kita natin. It's added after a while. Now, the editing will depend on, so essentially, no, the editing or the post translational changes within that are happening to polish, to finalize the sugar chain will be a combination of two events, no? Monomers being cleaved by from the chain via the action of hydrolase enzymes, ACEs, and monomers being added to the chain from a substrate containing them, usually nucleotide phosphates, via the action of transferase enzymes. Okay, so remember, ganun lang. Ganun lang yung nangyayari. Dagdag, bawas, dagdag, bawas. Eventually, finalizing the complex oligosaccharide, giving the protein, the glycosylated protein, a unique feature. Okay. Now, I already talked about the next slides. Yan. These are just notes to remember about the discussion I gave a while ago. Now, yeah, sugars. Some of the other sugars aside from the ones I've mentioned already: GLAC-NAC, mannose, galactose, sialic acid, fucose. Another kind of sugar, another kind of monomer sugar is NANA, N-acetylnuraminic acids. Remember that as another example of an important sugar monomer. Okay, that is added to um, the complex oligosaccharide chain. Okay. Now, what's the biomedical importance of this process, this terminal glycosylation? Yeah. There are a number of proteins that depend on glycosylation for their unique functions. Okay, basically these are the proteoglycans, the glycoproteins, which are heavily glycosylated proteins. They are required to form extracellular matrix. So you remember from your histology, diba, the ECM is actually one of the dominant components of the ECM are the proteoglycans. Proteoglycans are a special kind of proteins that are glycosylated, that are added with multiple chains of sugars. Now, also, aside from those making up the ECM, there are enzymes, acid hydrolases, that will rely on their modified N glycosyl groups for the targeting of proteins to the lysosome. I will talk about that in a little while. And antigens are essentially important proteins that are also glycosylated, giving them a unique characteristic or a unique feature. Okay. Now, okay, so yeah, so I mentioned about the enzymes, the transferases, the hydrolases, no? Okay, and aside from the enzymes involved in the process of adding and removing sugar monomers, and there are also other no no non what do you call this? There are also non-enzymatic proteins that are present in the GC, and you must remember, aside from the enzymes that are acting upon the growing complex oligosaccharide chain, there are also proteins that are embedded within the Golgi membrane, responsible for in a way, I don't know, tagging that component as a medial Golgi area, a cis Golgi area, or a trans Golgi area. Okay. All right. So now, we've covered terminal glycosylations. It's very important that we really include we top our discussion about glycosylation in proteins. No, from we as we started from the ER, then eventually leading to the Golgi complex. Okay, now other functions within the Golgi complex include protein trafficking. Okay, siguro ano natin, no? Yan, because we're done with terminal glycosylation. So, protein trafficking, protein sorting, and lysosomal formation. Okay, now let's first talk about trafficking or transport. Yan. Okay, so pathways that are important for us to remember. There is a secretory pathway. You start from the ER, then to the Golgi, then the secretory vesicles that will leave the Golgi area, and then the secretory vesicles fusing with the membrane, leading to exocytosis. And the cargo will be released to the extracellular environment. Okay. Also, there is an internal pathway or um, um, an internalization pathway. So, from endocytosis, okay, the process, we have our transport vesicles budding off from the cell membrane. Okay, so this actually you can call this the endosomal pathway. Endosomal, papasok ng cell. Okay, so maturation of transport vesicles of endosomes, okay, allowing the fusion of the delivery of the cargo primarily into the lysosome or the Golgi network or back to the ER depending on certain factors. Okay, now more about transport vesicles. Let's examine transport vesicles. Let's have a closer look at them. So as discussed, these transport vesicles are very important as the shuttling elements, the elements responsible for molecule flow. Transport vesicles they continually bud off from and fuse to other membranous components or compartments, producing the flux of materials that they carry. Okay, usually they carry soluble proteins. Those in within them or within the space, the vesicle space, no, are soluble proteins, while a lot of the non-soluble ones, a lot of the non-polar ones are embedded in the bilayer. Okay. Now, these transport vesicles, of course, their movement within the cytoplasm is made possible by the microtubules, by motor proteins that are embedded within the cytoplasmic space. And once that they recognize the target compartment where they will deliver the cargo protein inside them, fuse sila with that target compartment, with the target membrane. And the cargo protein within them will be released into the lumen, into the space of the target compartment. Yeah. The transport between organelles as mediated by transport vesicles involve special proteins that coat the vesicle. So these are coat proteins. Okay, so coat proteins. Some of the examples of coat proteins that are involved during organelle transport include clathrin, COP1, and COP2. Okay, now, so basic difference. Clathrin is associated to receptor-mediated endocytosis, internalization, the endosomal pathway. So expect clathrin as the coat proteins for the transport vesicles that carry material from the outside environment going inside. Okay? COP2 are coat proteins associated with the flow of material from the ER to the Golgi. So from the ER to the cis-Golgi network. Okay, transport vesicles are covered by COP2 proteins. On the other hand, COP1 are coat proteins associated with the transport of material from the Golgi back to the ER, or transport of material within the Golgi cisterna. O, tinatawag natin na intra-Golgi transport. The vesicles that shuttle the molecules are covered by COP1 coat proteins. Okay? So remember the difference between these three. Patrin, COP1, and COP2. Okay, so we have seen how vesicles, how transport vesicles act in the cell. No, meron tayong mga electron microscopy to as a technique to actually study that. So as an example here, you can see a vesicle budding, which is driven by the coat protein assembly. Okay, so here, eventually from the membranous component, from the membrane, the transport vesicle is uh, uh, excised, or in a way, ano siya, no? Nag-pinch off siya from the membrane. And you can appreciate the proteins surrounding the vesicle, the spherical transport vesicle. Okay. Okay, so I mentioned this already a while ago, the COP proteins. Okay. Let's take a closer look at the transport vesicles. Yeah. Aside from within the coat, no, the transport vesicle coated by coat proteins. Okay. You can expect the cargo to be arranged in a manner na it can be associated with additional proteins on the surface or on the outer layer of the transport vesicle. Okay, so here you can see the cargo in relation to coat proteins and in relation to the lipid bilayer. Okay, so usually the cargo will the cargo yun nga kung soluble siya, kung polar, kung tag dito, if it's very polar, then it will definitely not embed itself onto the onto the vesicle membrane. But if it's non-polar, may area siya, may segments siya that are hydrophobic in terms of amino acid residues. They can be, those components are most likely embedded within the lipid bilayer of the transport vesicle. So remember the difference between the COP. So COP2 coated vesicles, ER to Golgi. COP1 intra-Golgi or Golgi to ER. Okay? Yeah. Reminder lang. Okay, so covered this already. And then, let's see. Okay. Alright. Let's closely look at vesicle. Let's closely look at vesicle transport, no, as a multi-step process. Let's examine this one. Okay. So I have a good illustration here. Yeah. Okay, so we'll discuss all of this. Just give me a moment. Okay, so let's return to the recording. Yeah. Okay, let's take a look at vesicle transport as a multi-stage process. Yeah. Okay, let's take a look at the steps. Yeah. So basic things. First, the formation of the coated buds. Okay. So from the source compartment, source organelle or area, okay, the transport vesicle will pinch off, and then essentially it's composed of heavily composed of the lipid bilayer of that originating compartment, that lipid bilayer coated by the coat proteins. Okay, and the coat proteins will dictate where the transport vesicle will be delivered, no? Kung ano yung kanyang magiging recipient compartment or the target compartment. Okay. So originating donor compartment, target recipient compartment. Now, the transport vesicle from the originating compartment possesses a specific coat protein surrounding it that will allow the target compartment to recognize it. Now, the process of recognition will lead to docking and then eventual fusion of the transport vesicle and release of the cargo molecules inside the target compartment. Okay. Now, there are several or there are standing hypotheses regarding how target compartments recognize transport vesicles from donor compartments. No? Kasi the process is really easy to to memorize na from the formation of the coated buds to the coated transport vesicles finding their way to the target compartment recognizing them, and then allowing them to fuse and remove their coat, then eventually release their cargo protein. So, ganun na, napaka-simple lang ng proseso. But, of course, we have to look at how the process is actually happening. So, for transport vesicles, the targeting and fusion of transport vesicles, we have one very popular hypothesis, the snare hypothesis. Yeah. Okay. So let's examine this. This narrative. Okay. So according to the snare hypothesis for transport vesicle targeting and fusion, yan, the transport vesicle that is coated, allowing it to to move from one location to another. It's actually composed of there are additional elements that are associated with it. Okay, so some of those additional elements include a GTPase, a molecule called a V-snare. Okay, and then that's for the donor, no? Okay. And then for the recipient or the target, yan, elements that are found within the target membrane include a molecule called T-snare and tethering proteins. Tethering proteins that could be coiled coil in terms of nature or multi-subunit. Okay. Now, let's take a look at how things are happening during the snare in under the snare hypothesis view. Okay, so the proper vesicle is recognized and bound by the particular membrane anchored tethering protein. So it's the tethering proteins that are responsible for physically connecting the transport vesicle to or physically making the transport vesicle nearer, no, placing it nearer the target membrane, the donor membrane. Okay, the RAB GTPase bound to the incoming vesicle stimulates the association of the two snare molecules, the vesicle snare found in the donor membrane, to donor transport vesicle, and the target snare, the T snare, which is found on the target membrane. Okay, so with the association of the V and T snare, nagkakaroon ng membrane fusion. Okay, so the transport vesicle now will be fused with the membrane of the target vesicle or the target organelle. Okay, there's fusion, and the cargo will now be released to the target compartment. Okay, how the snare complex dissociates, additional elements are involved. The NSF and the SNAP proteins will play important roles in order to dissociate the snare complex, the V snare and the T snare complex. Okay, so if I am not mistaken, the snare hypothesis was first published in the year 1994. So it's more than ano na, no? Almost ano na siya, magti 30 years na siya, the hypothesis. Rothman, the scientist is Rothman. So Rothman is principal author, I think, of the snare hypothesis, no? And today we have so many things to understand about the snare hypothesis. Uh, it's a big, or I'd say it's an elaborate, very intricate no, process. A lot of people are working on. Okay. Okay, so I mentioned already, yeah, the elements, the T snares. So these proteins are complementary, so we have to remember that. So they recognize each other, okay, with the assistance of tethering proteins, so that docking will be successful, and eventually fusion of the donor vesicle, the donor membrane, will be possible with the to the recipient membrane. Okay. Now, there are different types of tethering proteins. No? Ano ko na to na I mentioned the V snare and the T snare already, so I won't elaborate on this slide anymore. There are different types of tethering proteins. So the two types generally are the coiled coil tethering proteins and the multi subunit tethering proteins. So let's take a look at them. The coiled coil tethering proteins, a very popular example, are the Golgins. They process the initial binding of COP1 and COP2 coated vesicles to the Golgi complex for the case of Golgin. So, coiled coil ang kanilang appearance, no? And at the same time as tethering proteins, they are also responsible for connecting cisterna of the Golgi network, of the Golgi complex. So para silang mga mini bridges that connect Golgi cisterna. Aside from acting as tethering proteins during transport vesicle movement, no, from donor to target. Now, another example, another subset of tethering proteins involved in the snare hypothesis is the multi-subunit tethering proteins. So some examples include COG, GARP, and TRAP. These are acronyms for specific multi-subunit proteins. No? And primarily these examples are important for the initial specificity and recognition of the vesicle target membrane interaction. Okay? Yeah, so yun. Okay, so we've covered transport, vesicle, or trafficking, molecule trafficking within the Golgi, or in particular yung transport vesicles, no? The idea of membrane trafficking by the action of transport vesicles. Now, there are instances pala, no, in connection with Golgi and ER as a continuum. Yeah. There are instances in which molecules that are intended for the ER are accidentally secreted away from it. No? So may mga ganong instances in which proteins that are supposedly found in the ER membrane, they're included in the transport vesicle na nasama sila, and then if not regulated, they will be lost, no? Or they will be they will move away from the ER. Yeah. Okay, so in such special cases of accidental spilling of proteins from the that are expected to stay within the ER or the Golgi, that accidental spilling can be prevented or can be counteracted via the process of retrograde transport, no? Which involves, of course, the transport vesicles, the snare mechanism, and in addition, signal siya, no? It's signal dependent. So there is within the cargo itself, let's say the cargo being a polypeptide intended to stay in the ER. So that cargo will have a specific molecular signature, usually no, in the form of amino acid sequence that will dictate na it should be within the ER and it should not accidentally spill away from the ER. So yung tinatawag natin na Cadel sequence. So later I will talk about that. Let's complete our discussion of protein trafficking and secretion. No? The last uh, the last questions we have to try to really give a resolution are ito, no, how are proteins sorted to appropriate vesicles so that they are transported to their proper destinations or proper location. Now, how are our address labels given? I think in our synchronous session for module 5, I already talked about signal sequences, no? For ER, for mitochondria, for the nucleus. So these signal sequences are important. No? Okay. Aside from actually heavily dependent sa signal sequences ang sorting, ang process of sorting within the cell, no, where the in connection to how the Golgi facilitates the process. Okay, now two pathways I already talked about, the internal and external. I will also have to look at pathways of protein secretion, of molecule secretion, in the context of um, answering for a need or answering for a signal. Okay, so one of the two pathways called constitutive is an unregulated pathway. So in constitutive secretion, the cargo is transported from ER to Golgi to outside the cell in a fashion or in a manner that is not signal dependent. So you can say that there is no cue, no cue for or no signal na that molecules needed to be secreted. So constitutive, it's done every time. So it's happening, whatever happens or whatever is happening in the cell, it's always done. So that's how constitutive pathway is. Um, it doesn't require a cue or a signal in order for it to take place. On the other hand, okay, the secretory pathway that involves uh, uh, a regulated no, rhythm or regulated direction, a regulated, yeah, in a way, no, regulated transport direction. Okay, so for this pathway, regulated secretion, there needs to be a signal, a cue that the cell recognizes to be present within it or outside in order for the secretion to take place. So, as an example, we have um, pwede tayong ano ba, mga sabihin natin the release of insulin. Okay, so when the cell recognizes sugars, glucose, no, they're glucose receptors, no, for that are processed by certain cells. So when cells recognize that, when insulin secreting cells recognize the presence of glucose in the extracellular environment, and that's the time that they will be secreting necessary molecule to counteract the process. So here you can see, um, secretory granules that contain insulin. For the case of pancreatic cells, they undergo regulated secretion. Okay, so when there is so much glucose in the blood, then that's the time that they get to secrete insulin. Okay, so that's the regulated pathway. So there is control in terms of molecule transport, molecule secretion. Okay. Ayan, ito. Okay, so let's go back to retrograde transport. Okay, so ano na, we're blurring the lines between transport and sorting, okay, or transport and address labeling. Okay, so if secretion is default, how are resident ER proteins retained? No, so in the process is actually the thing is ER proteins are not really retained, are not really actively kept. So there is no inhibitory mechanism that we know to date, no, that prevents ER proteins from being accidentally spilled from the ER region to the Golgi and towards secretion, no, in order to retrieve yung ER protein. So the verb is retrieval, okay, or recycling. Okay, there are numerous elements, numerous components. We have yung Cadal sequence, okay, so that Cadal sequence found in the C terminus of the protein, okay, serves as a very important a molecular signal telling the organelles uh, to retro transport the ER protein or the protein to the ER because it's supposed to be in the ER and not within the Golgi. So pabalik, no? Sample, very important yung role ng Cadal. So the Cadal motif. So K, lysine, D, aspartic acid, E, glutamic acid, and L, sorry, wrong spelling. This should be leucine. Okay, let me just correct. Okay. Now, all right. So let's now summarize everything before we proceed to lysosome targeting, to lysosomal targeting. Okay. So okay. So saan tayo mag-start? Okay. So here. So protein targeting. Okay. So within cytoplasm, you remember co-translational import and post-translational import. Okay, so signal sequences are important. Okay, ER signal sequences are recognized, and the proteins that are translated will either be imported to the ER prior to translation being finished or while translation is happening. Okay, and then there are, aside from the signal sequences, marami tayong klase ng signal sequences. Those that recognize the need for the protein to be embedded within the ER membrane, the need for the protein to be translocated in the ER lumen, or certain components to be exposed in the cytoplasmic space. Okay, so default is eventually the proteins from the RER, the rough ER, will be transported to the Golgi. Okay, now for the case of soluble proteins, the Cadal sequence is recognized as a signature, so that if ever there are proteins for the ER that are accidentally spilling to the Golgi and towards the secretory pathway, maibabalik sila. for soluble. Now, for the case of the grounded or the embedded proteins, the membrane proteins, the KKXX sequence is the KKXX motif is recognized. Okay, then for as the protein travels within the Golgi, default is constitutive secretion. The transport vesicles will fuse with the plasma membrane, secreting their contents. Okay, then we also have regulated secretion, secretory vesicles requiring signals, no, following a signal-dependent pathway. Without the signal, the vesicles will not fuse with the plasma membrane. Okay. Now, from the Golgi, since we know, diba, that from the Golgi, the lysosomes, the lysosomal vesicles are produced. How are proteins targeted to the lysosome? Okay, so we have a few things that we can remember, that we can always go back to whenever we study how proteins are targeted to the lysosomal space. Yeah. Proteins that are of acid hydrolase nature, the ones that are degradatory to biomolecules, okay, these proteins are modified with mannose 6 phosphate residues. So you can think of mannose 6 phosphate as an important molecular signature telling that the protein should be within lysosomes. So it's a signature of acid hydrolases. Now, aside from mannose 6 phosphate as a signature, there are also soluble enzymes that are targeted to the lysosomal space and also non-enzymatic proteins that are following the M6P independent targeting mechanism. So some proteins, such as those that are facilitated to be those that are targeted via the action of proteins such as sortilin and LIMP2. So these are M6P independent pathways, but eventually this too will allow soluble enzymes, non-enzymatic proteins to be targeted to the lysosomal space. Okay. Now, sorting signals, aside from mannose 6 phosphate, that can also allow the Golgi to really compartmentalize as a biomolecule to the lysosomal components. Lysosomal vesicles include dileucin motifs and tyrosine motifs. Okay, so aside from the sugar. So meron kang sugar sequence, meron kang sugar monomer signature, meron ka ring amino acid signature. And there are also other pathways that are independent of sugar. Okay, so there are pathways that involve special proteins that will target the final protein to the lysosomes. Okay, so that's the last slide. That ends our discussion about the Golgi complex and lysosomes. So we've covered discussions on the nature of the Golgi, what's happening in the Golgi, and the important processes that the Golgi carries out. Okay, processes of terminal glycosylation, protein molecule trafficking, protein sorting, and protein targeting. Okay? As well as i-blur din natin, include din natin of course yung secretion. No? It's important function that the Golgi is taking care of. Okay, so that's it. It's the last slide for this module. So we close module number 5, no? The endomembrane system discussion with the Golgi complex and lysosomes. So please make sure that you will watch all the recorded lectures to help you be capacitated in answering our enabling assessments and also the summative assessment.